Hi everyone, my name is Emma Rauschenbach, and today I'll be presenting an investigation of the effects of plastic ingestion in juvenile Hawaiian wedge-tailed shearwaters. So as many of us know, plastics are a major issue for marine ecosystems. Through improper disposal and a lack of management, they've become one of the most abundant materials to appear as marine debris. They're extremely hard to monitor. They're typically free-floating, which means they can travel extensively, and they're durable. While many of them will break down into secondary plastics or microplastics, they can never truly degrade. Until now, these challenges have been met using advanced technologies that are costly and unsustainable. However, recent studies have indicated that seabirds have the potential to be useful bioindicators as a low-cost and a sustainable alternative. All things considered, my main motivation for this study was to investigate plastic consumption in wedge-tailed shearwaters as well as their potential as bioindicators. For this project, we necropsied 107 wedge-tailed shearwater chicks from 2011 to 2014 and collected both of their stomach chambers, the proventriculus, and the gizzard, giving us a total of 214 samples. When sorting these samples, we looked for three categories of ingested materials: natural foods such as small fish or squid, natural non-foods such as twigs or sand, and plastics. To be a successful bioindicator, a species needs to have some level of compliance with non-invasive sampling techniques such as regurgitation sampling. So by understanding the retention and effects of plastics on wedge-tailed shearwater diets, we were also able to evaluate their utility as bioindicators. Wedge-tailed shearwaters are an endemic species to Hawaii, and their scientific name was recently amended to Ardenna pacifica. They are multi-trophic marine-feeding seabirds who utilize various feeding techniques, including contact dipping, surface seizing, and plunge diving. Their diets typically consist of squid, crustaceans, small fish. However, in recent years, this has come to include plastics as well. Ingested debris, as illustrated on the right, often includes consumer items such as bottle caps, floss, fishing line, and miscellaneous microplastics. My research investigated 4 hypotheses and goals. I first predicted that plastic ingestion would vary between years given the La Nina event that occurred between 2010 and 12, potentially influencing plastic availability in offshore feeding environments. I also predicted that plastic incidence, size, and mass would vary between stomach chambers. As you can see on the right, the proventriculus is a thin-walled, smooth muscle chamber that sits right above the gizzard, which is much smaller and more rigid. Given its size, elasticity, and location, I predicted larger plastic items with less incidence would be found in the proventriculus, while more plastic items, smaller in size, would be found in the gizzard. And finally, I predicted that natural food would decrease as plastic mass increased, and that fragments would be our most abundant debris type. All of our samples were opportunistically collected by the Sea Life Park and the Oceanic Institute between 2011 and 2014. They were kept in a -9°F freezer until necropsy, where their stomach chambers were then separated and stored in a 30% ethanol solution. We used standardized necropsy procedures and verified all of our birds to be chicks. To sort ingested materials, I first placed each of the stomach chambers over a 300 micron sieve, rinsed them with a 30% ethanol solution, placed them on a petri dish to dry, and then sorted them under a stereo microscope into each of their respective categories, which were then weighed on a digital precision scale in grams to the nearest 10,000th. So plastics were first identified by observation and then pre-verified using a float test, which included placing suspected material into a beaker with water, and if it floats, it's probably plastic. If it sank, it was organic. And then I used a biological indicator dye called rose bangle to separate the remaining organic material. Plastics were then analyzed using a micro Fourier transform infrared spectroscopy, or FTIR, where their size, weight, debris type, color, and polymerization, either high or low density polyethylene, was recorded. Analyzing total number of plastic items as a function of year, I found that there was a significant decrease in ingested plastics. A Kruskal-Wallis test was used to identify an overall significance, and I ran a Wilcoxon test to identify that significance was between 2011 and 2013. For my second hypothesis, I looked at the number of plastic items in each stomach chamber, abbreviated PRO or GIZ, for each year. A Kruskal-Wallis test was used to determine overall significance, and individual Wilcoxon tests were used to determine significance in the years 2011 and 2012, as well as 2013. For size of plastics, I first used a regression model to verify length as an appropriate variable of size. I then used a Kruskal-Wallis test to determine overall significance of size between each of the stomach chambers, and it turned out that the proventriculus held significantly larger items than the gizzard. I then looked at plastic as a function of each stomach chamber and used a Kruskal-Wallis test to determine that mass was significantly greater in the proventriculus than the gizzard. I also investigated if natural food was affected by plastic ingestion, and after running a linear regression, I found there was no significant correlation. When birds ingested less food, they ingested less plastic, and vice versa. I found 2 out of the 4 types of plastics we looked for to be present in our samples, fragments and line. I used a Kruskal-Wallis test to determine the significance as well. Overall, we found that most likely due to the La Niña event of 2010 and 12, there was significantly more plastic in 2011 and 13. This could be because as the event ended, the currents subsequently pulled the plastic away from offshore feeding environments, resulting in a lower amount of plastic in 2012 and 14. We also found that in years with more plastic, the incidence of plastic in the proventriculus was significantly higher than in the gizzard, whereas in years with less plastic, the gizzard had a higher incidence. This is most likely an indication that the gizzard has a higher retention of plastic, so as plastic started to leave feeding environments in 2012, their incidence remained in the gizzard and subsided in the proventriculus. The proventriculus also retained larger plastic items and had greater mass of plastics than the gizzard. We also found that there was no significant correlation between natural food and plastic ingestion, possibly due to our sample size, and finally we verified that fragments were the most abundant debris type. In conclusion, I found support for 3 of my 4 hypotheses. Natural food as a function of plastic was determined to be insignificant with the sample size. However, I found that plastics significantly varied between years, size, mass, and incidence significantly varied between chambers, and the most abundant type by far was fragments. Using chicks, we only analyzed the effects of secondary plastic ingestion. To improve significance of our findings, future research could include a larger sample size as well as age variation to illustrate primary consumption patterns. Since we have established that there is a significant difference between the retention of plastics between stomach chambers, we've also helped to establish their effectiveness as bioindicators. My acknowledgements to my mentor and grad student Sarah Hutchinson, as well as graduate Timothy Alonzo. Thank you.